Hello everyone. Welcome to today's section. The topic is factors influencing microbial growth, environmental and nutritional. Myself, Jumana Karadan, second MSc biology student, St. Mary's College, Thrissur. First of all, we have to discuss what is microbial growth. Microbial growth is orderly increase in quantity of cellular constituents and number. It depends upon the ability of the cell to form new protoplasm from nutrition available in the environment. The increase in bacterial cell number occurs by cell division, which is known as binary fission. In case of bacteria, binary fission is the microbial growth of. So binary fission begins when DNA of the cell is replicated. Microbial growth phases. Microbial growth have four phases. These are lag phase, log phase, stationary phase and death phase. First of all, lag phase. During lag phase, there is no increase in cell number but an increase in mass. Thus, in this phase, the microbial cells are not dormant. The length of the lag phase is determined in part by the characteristics of the bacterial species and in part by conditions in the media. The second phase is log or growth phase. Here the cell number increases exponentially with time. Log phase, also known as exponential phase. During exponential growth, the rate of increase of cells in culture is proportional to the number of cells present at any particular time. The rate of exponential growth of a microbial culture is expressed as generation time. During the exponential phase, each microorganism is dividing at constant intervals. Thus, the population will double in number during a specific length of time called generation time or doubling time. So, we have to calculate generation time. Let N0 is the initial population number, Nt is the population at time t, N is the number of generations in time t. Then Nt, that is the population at time t, which is equal to N0 into 2 raised to n. The generation time, which is equal to t by n. Microbial growth curve. The graphical representation of microbial growth phases known as the microbial growth curve. Here we can see the four phases of microbial growth: lag phase, exponential growth phase, stationary phase and death phase. Lag phase, here we can see few live cells are present. Exponential growth phase have many live cells present. Stationary phase have many live cells and also little dead cells. And the death phase have more dead cells. So the third phase is stationary phase. Exponential growth cannot be continued forever in a batch culture, that is a closed system. In this phase, cell growth rate has leveled off and become constant. The number cell multiplying equals the number of cells dying. And the fourth phase is death phase. If incubation continues after the population reaches stationary phase, a death phase follows, in which viable cell population declines. The number of viable cells decreases geometrically because the nutritional availability in this culture media decreases as the population grows. So it reaches to the death phase. So now we can discuss the main topic: influence of environmental factors on microbial growth. Growth of microbes is greatly affected by the chemical and physical conditions of their environments. Many environmental factors like temperature, pH, water availability, and oxygen play major roles in controlling the growth of microbes. Plasmolysis, which is a factor, is a kind of a factor, that is dehydration of the cell in hypertonic environments may damage the cell membrane and causes the cell to become metabolically inactive. Microbes in hypotonic environments can reduce the osmotic concentration of their cytoplasm. This can achieved by the use of inclusion bodies. Some bacteria and archaea also have mechanosensitive channel in their plasma membrane. So plasmolysis is a kind of factor which influences the microbial growth. The other one is water activity can be represented with the AW. It may be expressed as water potential. The water activity of a solution is 1 by 100 the relative humidity of the solution. It is also equivalent to the ratio of the solution's vapor pressure to that of the vapor pressure of pure water. That is water activity which is equal to the ratio of vapor pressure of a solution to vapor pressure of pure water. A microorganism must expend extra effort to grow in a habit with a low water activity value because it must maintain a high internal solute concentration to retain water. Some microorganisms can do this and are osmotolerant, that is, they grow over wide range of water activity. Example, Staphylococcus aureus. So water activity is a kind of factor which depends upon the microbial growth. So microbial response to environmental factors. Many description terms are provided with the definitions and representative microorganisms. So first of all, solute and water activity. The term is osmotolerant, which means able to grow over wide range of water activity or osmotic concentration. Example, Staphylococcus aureus. Halophile, which that means require high level of sodium chloride, usually above about 0.2 molarity to grow. Example, Halobacterium. The second one is pH. The term acidophile, which means microbes have a growth optimum between pH 0 and 5.5. Example, Sulfolobus. And other term is neutrophile, which means growth optimum between pH 5.5 and 8.0. Example, Escherichia. And the other term is alkalophile, which means growth optimum between pH 8.0 and 11.5. Example, Bacillus alkalophilus. And the third one is temperature. Temperature is a kind of factor which depends upon the microbial growth, and so many terms which are related to temperature. Those are psychrophile, which means grows at 0 degrees Celsius and has an optimum growth temperature of 50 degrees Celsius or lower. Example, Bacillus psychrophilus. The other one is psychrotrophs, which means can grow at 0 to 7 degrees Celsius, that is has an optimum between 20 and 13 degrees Celsius and a maximum around 35 degrees Celsius. Example, Pseudomonas fluorescens. The other term is mesophile, has a growth optimum between 20 to 45 degrees Celsius. Example, Escherichia coli. The other term is thermophile, can grow at 55 degrees Celsius or higher. that is optimum often between 55 and 65 degrees Celsius. And the last term is hyperthermophile, has an optimum between 85 and about 113 degrees Celsius. Example, sulfolobus. And the fourth term is oxygen concentration. Obligate aerobe, which means completely dependent on atmospheric oxygen for growth. Example, Micrococcus luteus. The other term is facultative anaerobe, which means does not require oxygen for growth but grows better in its presence. Example, Escherichia. Then aerotolerant anaerobe grows equally well in presence or absence of oxygen. Example, Staphylococcus pyogenes. Then obligate anaerobe does not tolerate oxygen and dies in its presence. Example, Clostridium. Then microaerophile requires oxygen levels between 2 to 10 degrees Celsius 2 to 10% for growth and is damaged by atmospheric oxygen levels, that is 20%. Then the other factor is pressure, barophile, which means growth more rapid at high hydrostatic pressure. Example, Photobacterium profundum. Here we can see some kind of bacteria which are osmotolerant kind and also halophile type. Here it is the graphical representation of rate of the growth to the temperature. How the temperature depends upon the microbial growth can be understand by this graph. And the second major kind of factor which influence the microbial growth is nutritional factors. Nutrients are the substances used in biosynthesis and energy production and therefore are required for microbial growth. All bacteria require several macro and micronutrients. Macronutrients, that means which are needed in relatively large quantities. Example, carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium and iron. And the second one is micronutrients, which means these are used in very small amount. Example, manganese, zinc, cobalt, molybdenum, nickel, copper. In addition to the need for carbon, hydrogen and oxygen, all microorganisms require source of energy and electrons for the growth to take place on the basis of sources, carbon, energy and hydrogen or electrons. Micros can be categorized into following types. The first one is based on carbon sources. The carbon requirements of the microbes must be met by carbon dioxide or organic carbon. Microbes that uses carbon dioxide for growth called autotrophs. Microbes that uses organic carbon are called heterotrophs. The second one is based on energy source. Microbes that uses light as their energy source are called phototrophs. Microbes that obtain energy from oxidation of chemical compounds are called chemotrophs. And the third one is based on hydrogen or electron sources. Microbes uses reduced inorganic substance as their electron source are called lithotrophs, whereas microbes extract electrons from organic compounds are called organotrophs. So the major nutritional type with the examples: Photolithotrophic autotrophs, carbon, energy, electron. Example, purple and green sulfur bacteria. Second one is photoorganotrophic heterotrophy. That means carbon, energy, electron. Example, green non-sulfur bacteria. And the third one is chemolithotrophic autotrophy. Carbon, energy, electron. Example, nitrophine bacteria. So here we can see some kind of microbes. Those are nitrophine bacteria and green non-sulfur bacteria. So we can expect some kind of questions from this microbial growth section. For 1 credit, we can expect explain major nutritional types with example. For credit 2, explain growth phases with a graphical representation. For credit 5, write a detailed description about factors affects microbial growth. That's all, and thank you.